on that.
Welcome everybody to today, and thank you for joining us. My name is Ben Grave, and I'm a surgeon here in Adelaide and also a board member of Craniofacial Australia. And we have a very nice group, and please feel free to use the toilets, and the toilets are just out through the doors next to the lifts. Also feel free to bring some food in here, and after our meeting this morning, especially the formal part of it, you'll be able to ask some questions as well to our researchers. It's a good morning because we get a chance to step behind the scenes to see how your support, that's very valuable to us, helps drive groundbreaking research discoveries for craniofacial community. At Craniofacial Australia, we rely on the generosity of donors and supporters to continue our work. This provides life-changing care, support, research and educational programs for our craniofacial community here and also overseas. Today we are celebrating our research arm. The scientists are working very hard to prevent and provide background information to how we can treat our patients better. These researchers rely on our grants, made possible through donations, and that helps us to really fund our understanding of how we can improve our care for patients. So please join us and welcome our researchers. Firstly, just to the right of me is Associate Professor Quentin Schwartz, who's a developmental neuroscientist. Associate Professor Seb Dawkin, who is a developmental geneticist, and Dr. Amanda Osborne, who's a clinical neuropsychologist. So thank you very much for coming. Also, I'd just like to welcome Associate Professor Neil McLean, who is from the north of England and is a very senior plastic and reconstructive surgeon, has been a great friend and colleague of Professor David over many years, and he's here as a board member and also as a very good researcher as well. So thank you, Neil, for coming. So we're going to divide it up into three parts. Firstly, to go through each of our researchers and to ask questions, and then finally open it to the floor to ask some questions at the end. Is that okay? So our first speaker is Quentin. This is his home here. This is the building that he works in, and he's from the University of South Australia and the Centre for Cancer Biology. We're very fortunate to be able to visit his lab today, and his associates, Kay Lee and Raf, will help take us through the labs afterwards, and please take your time and ask as many questions in that part as well. During the lab tours, you'll have a chance to see some of the imaging platforms, the techniques and tools that they use day to day. Quentin's research focuses on very complex, but my understanding that there is an interest in the genes and how those genes are controlled and how they're controlled in the development of the head and face. This is a short video, first of all, just as an introduction, and then we'll hear from Quentin himself.
Our major interest is in trying to understand how do craniofacial defects arise. Our hope is that by understanding how they arise, we can someday alleviate that, stop it before it even forms. What we're trying to do is add to that repertoire of expertise in the state by adding to that knowledge of how the face forms, how then treatments could be improved so that we have better outcomes for kids. So my name's Quentin Schwartz and I work at the Centre for Cancer Biology. I run the Neurovascular Research Laboratory, and our aim is to try and understand the fundamentals of how the cells that give rise to the face and the cranium, how they form, how they get to where they should be, how they then mature into cartilage and bone, which is what the structures of the face are. So going back to what we do in the lab is try and understand how development of the face forms during embryogenesis. And what we try to understand is how the cells mature in such a way that they can give rise to the structures of the face. That could be the jaw, it can be the skull as well. When craniofacial disorders arise, it comes about due to a fault or a problem during that normal developmental process. So in this case, the way that the bone cells form, they mature and they come together. Our understanding from the current project will have impacts across craniosynostosis, cleft lip and palate, and also to more syndromic type of cases. Our work strives to understand how the bones and cartilage form in the first place, and then try and come up with ways of changing how the bone and cartilage forms so that that could lead to new therapies. If you know how to put a car together, you pretty much know how to fix it. All right, so the idea is that if we understand how you put all of these elements together, then it will be easier for us to understand how it goes wrong. And then by understanding those things together, we can come up ways of stopping or fixing it. I'm very, very grateful of the support that I've had from Craniofacial Australia. This is the second grant that we've had from Craniofacial Australia over the last 3 or 4 years. And that funding has allowed us to undertake pilot studies. And what's been really great about that is those pilot studies, along with input from Professor David, have allowed us to then use the pilot studies and background data to apply for larger funding in the sense of time, but also the amount of funding. At the end of last year, we're really fortunate to get National Health and Medical Research Council funding, which is now going to allow us to continue with the work that Craniofacial Australia has funded for the next 4 years. Katie Marchant from my team is championing all of the work at the moment in the lab around the current work that's funded via Craniofacial Australia. And she's leading projects where she's looking at how cartilage cells are able to survive. And we use a lot of models for this type of research. In this case, we're looking at cells in a dish. And so what Kaylee is looking at on the screen, separate cartilage cells that we can then manipulate in different ways to understand how they make more cells or how they survive. And what Kaylee is able to do with these is add different factors to the cells to make them turn into cartilage, but also to turn into bone, but also under stress how they die. And that's one of the projects which we're currently working with Craniofacial Australia on. Our vision in the lab is that we work together with the families and the clinicians to try and get better outcomes. We're not going to do that overnight, and it's really important that we just keep striving together. What really excites me is that we're at the cutting edge of it. On a day-to-day basis, we're finding out new things that no one else in the world knows. If we can come up with better ways of treating and better outcomes for patients, that's our why. We want to be able to stop it in the first place and make the outcomes better.
Good, thank you very much. That was very, very interesting. So the first question, Quentin, we know that when the face develops in early embryogenesis, there is the neural crest cells that really come down. And we know from our clinical work that sometimes that causes facial deformity. Now, you're going down in the lab looking at all of those aspects of development. How does that help us get better outcomes for patients?
Sure. So, well, welcome everyone here today. I just want to touch, before I answer that question, you said thank you to the researchers for coming here today, not far from me. Just to point out that Seb's driven all the way from Melbourne, so 8.5-hour drive. So we're very thankful he could do that as well. And you've come over from University of Adelaide, so correct. You know, still had to get here. I had to just come downstairs. Anyway, proof of the pudding there that you saw the video that editors can do amazing jobs with things. You've now got me in real life, and you're going to have to wrestle this microphone off me at some stage. Answering your question, probably acutely aware that the audience is not scientists. Really hard job for us is to try and keep the jargon out of what we're saying. And so if I'm starting to not make sense, just shut your eyes and I'll pick up pretty quickly. All right, so neural growth cells. Love these things, right? This has been a passion of mine since I was an undergraduate student. Every chance I got throughout university, which I did here in Adelaide, was on projects based around neural growth cells. I did my PhD on the periphery of neural growth cells. I then moved to London. I was there for 6 years, where I got a foundation in developmental biology, and that is the area of how does a single fertilised egg go through embryonic development to give rise to something that's a relatively normal child. It goes wrong a lot of time, and my lab focuses on that. So we know that craniofacial defects can arise. One of the ways they can arise is because neural crest cells, this is a progenitor cell type of all the cartilage and bone in our faces, right? So if they don't form, we don't get the proper structures. Hence, you'll have a craniofacial defect. There are many other ways though. So my PhD mentor, Timothy Cox, which I think you might know, Ben, I know Neil will know him. He focuses on craniofacial defects from another perspective, which is the epithelium. That's the skin, right? So how does the skin during development communicate with the neural crest cells to create the structures? And I think you'll probably hear from Seb about that as well. Yes, we are very focused on neural crest cells, and one of the things we know is that neural crest cells are formed, then they have to do a billion different things to actually give rise to the structures, and then to be in the right spot to then talk to other cell types. So for us, our lab has a major project at the moment where the neural crest cells start to form cartilage and then die, and we don't know why. Right? We have genetics that tells us why that is the case. We have other projects where we know neural crest cells don't move to the right area. So, you have to get from, I had to get from level 10 to get to level 8. If I couldn't do that, I wouldn't be here, right? So then I can't talk to you today. Same thing happens with neural crest cells. They need to move from where they are to somewhere else to give rise to those structures. So what we are really pushing towards is, if we understand how neural crest cells don't give rise to bone and cartilage, if we know the mechanism, so that's molecular mechanisms, then we have ways of trying to stop that from occurring. So I mentioned Kaylee in the video, and also Rafa Katz over here as well. He's a PhD student in the lab. Their particular project is looking at how neural crest cells will die. And we've got some really good evidence now that we know why they die. As I take you through the labs later, you'll see part of why we know that. It's because the cells become stressed. And that stress comes about because they don't get enough nutrients. So hence, and I think, Ben, you may have seen this, we can add extra nutrients to these cells and it keeps them alive. And not only does it keep them alive, it actually protects the outcome of the craniofacial defect. So I think what we're trying to aim at, where we're trying to get to, is, can we come up with simple ways? Could a mother change her diet somehow to protect the child in the womb, and so that wouldn't have as a severe outcome in a craniofacial defect at birth? Now, that would be impressive. I know Seb's working on this as well. That's the holy grail of the research, and that's what we're, the big vision. And like I said in there, it's not gonna happen overnight. We are getting really small steps towards that, and I think I'll just tell you one of the outcomes of the work is that Kaylee found more recently. We do a lot of our work in cell models, and that's because we can, and it allows us a quicker time frame to get to answers. But we do have to do some of our work with animal models because it's a complex structure, right? There's all these three-dimensional things that have to change. And in one of our animal models, we can simply add more leucine in their water, and that can protect against the craniofacial defects. I'm not saying that could happen to every individual, but if you think about the addition, now can you help me out here, to stop neural tube defects?
Yes.
So folate, just by adding folate to cereal, how amazing effects that's had for birth defects. And I see a future where we can do this for craniofacial defects.
Just going on from that, Quentin, just with the idea that there is a craniofacial deformity, so there's a bone deformity, cartilage, bone, soft tissue. Is there a connection between some of the neural crest defects and brain development? And have you seen that in some of your work here in the lab?
Yeah, 100% we have, yeah. So a lot of the models we use to look at craniofacial defects arise because we've made genetic changes. Right? So a genetic defect, which could be inherited from a mum and dad, but it not always the case. It can just happen randomly. And I think the bulk of these times we cannot predict from mum and dad's genetics if the child would have a defect. Right? It just happens, unfortunately, while the embryo is forming. Now, you asked me about the neural crest and the brain. So these two systems are heavily connected. And there's another PhD student in the back here, Matt, who can probably raise his hand, and his entire PhD project is looking at that. How are the similarities between the brain and the cranium entwined? How do they talk? How does it actually come together? But I think it brings up a bigger problem, right? It's because we know that a craniofacial defect will have its own challenges. It will lead to difficulties in some parts of life, whether that's breathing, whether it's eating, and other things. But we also know that, unfortunately, if the top of the head is fused too early with craniosynostosis, then there's no room for the brain to grow. But on top of that, there's also potential problems that the brain cells don't form properly either. And that could just be, unfortunately, because the same genetic change leads to that. If we can understand those things better, we can also affect other areas that are problems for the children.
This is very vital, and Dr. Amanda Abbott will elaborate on some of that as well a little later. Now, I thought it good just to move on to Seb. And as mentioned, Seb is from La Trobe University and has travelled by car, really, to be with us today, and also on Sunday as well. And thank you very much. And Seb leads a pioneering research into genes and cell shape and developing aspect of the jaw and face as well. So his work is obviously supported by our foundation and is very revealing and has been very impressed by us as a board, the outcomes that has occurred. So let's watch a short video first and then ask him some questions, and we'll hear from him directly.
I'm Seb Dworkin. I work at La Trobe University in Melbourne, and my work is largely funded by Craniofacial Australia, who are really a very special charity who do exceptional work in making lives better for children affected by craniofacial conditions and their families. And without their critical support, we would not be able to do the important groundbreaking work that I'll be telling you about. Would you believe that at the moment we don't know what causes about half of all craniofacial malformations or craniofacial differences? And so what my group is trying to do is try and uncover some of those mysteries by investigating the genes that are responsible for these differences in development. So one thing, for example, we've been working on is trying to understand how vitamins and minerals and changes in diet can impact on embryo formation. If I was to say to a mum, here, take this medication that I've developed during pregnancy, they'd be hesitant. If they can make small changes, for example, we already know that things like folate are important and iodine. What excites me is trying to identify the next beneficial, healthy vitamins that we can get from our diet to again improve the start of life for our children. And we also study relatively more common conditions such as cleft palate, which is a gap in the roof of the mouth, and a condition called micrognathia, which sounds a bit complicated, but all it means is a small jaw. If you only remember one thing about your donation, know that it is directly impacting the lives of hundreds of people. It directly funds critical experiments, it directly pays for our wonderful researchers, and it helps fund student projects to not only help them develop their careers, but for them to get passionate about craniofacial research and then take that information with them as they start on their own lives. Way back when I first started my work following my PhD, I was actually more interested in the formation of the brain and the cells that form the brain. But when I was using one of my models, normally the brain develops at 24 hours. But one day, it was a Friday, and I thought I'll just wait a few extra days and see what happens. Now, the brain develops in 24 hours, but the craniofacial skeleton doesn't start to form until about 4 days. And so I thought I'll just leave this over the weekend and I'll come back on Monday and have a look at it. And I had a look, and to my incredible surprise, I found that the whole lower jaw was missing. And I thought, this is incredible. Why has this happened? It was a very unexpected finding. And it really has set up my research career from that one moment, because the more I investigated, the more I realized that other people had also seen that this gene that I was working on was involved in formation of the face. And so that was about 15 years ago. And since then, I've just been fascinated by how the pieces of the puzzle that make a face fit together and how the genes, the master regulators, actually control this. The more I got to know people at Craniofacial Australia, the more I realised the impact and the need for the kind of work that I'm doing. A previous research grant I had from Craniofacial Australia allowed us to do some key work that found that the way the palate closes, as well as the neural tube, which is the tissue that makes up the brain, the way that closes, was that the genes involved in both these processes were actually the same. The work in my lab will directly go straight to the doctors who are dealing with families. And the better informed that the doctors and the genetic counsellors are, then obviously the more comfort and support they can provide for families. So ultimately, that is my goal.
And thank you, Seb. I had a chance to have a chat with Seb before, and he just helped me understand his work. And one of the things he said was he literally knocks out a particular gene in an animal model and then watch and see what effect that has on the end result, what for deformity arises. Now, Seb, could you let us know how accurate that is in the human population? And because we know that a little child born with the conditions that you're looking into are very various and quite significant clinical scope.
Yeah, well, that's an excellent question. And firstly, I'd just like to thank everyone for coming, and thank you to Craniofacial Australia for letting me join this panel. I was very, very happy to join. No, that description is completely accurate, actually. So one of the key things we do, and you would have heard right at the very start of that video, is that at the moment, we don't really know what causes most of these craniofacial defects. And as you can imagine, understanding those genes is really a critical part, both for diagnosis and to give families and children certainty as to what's causing their condition, but also to then try and intervene. So much like what Quentin was saying, one of the questions that really drives me is, what can we do before birth to minimise the impact? Now, obviously, the post-birth surgical care is excellent, but I'm always thinking, if we can do something to limit the severity and limit the need for that kind of surgery, then that can only be a good thing for the children. So my lab takes the approach of deleting certain genes, and one of the models we use is zebrafish. I should have also said I'm not going to be anywhere near as eloquent as what Quentin was, so I was very impressed. And now he's gone. But yeah, so we use zebrafish, for example, and you heard from Quentin about neural crest cells, which are very, very important in forming the face and the craniofacial skeleton. But what my lab really looks at is not so much those cells themselves, but how they're organised and how they're driven to form the bone and the cartilage. And an analogy you might know, you might have seen the ads for Carlton Draft and National Australia Bank. I'm not sure if you had them here in Adelaide, I assume you did, where you have those hundreds and hundreds of people all coming together and forming a big star or forming a big beer glass. And that's, in a way, like the neural crest cells. Yes, you need those neural crest cells, but you need some instruction. You need to give them some instruction for how to form that structure. And what my lab looks at is the tissue that gives those instructions. And so we find that if we make deletions in genes that regulate that structure, it's actually called the pharynx, which you might have heard of, we can then impact on how the neural crest cells are instructed and how they're regulated. And so you can imagine if those people didn't have the instructions to form a giant star, for example, they wouldn't do it properly. They'd still be there, but they just wouldn't know what to do. And so one of the cool projects we have going at the moment is that we found that in our zebrafish, depending which environment they're housed in, they have either more or less severe craniofacial deformities. And I thought this was actually quite incredible because we had the fish at 3 different universities in Melbourne, and we found the outcomes were very different based on the water quality and based on actually the microbiome they have in their stomach, the bacteria in their stomach. And so this is actually quite new. This is we only really discovered this in the last sort of 6 to 8 weeks. So it's really quite cutting edge. But what we're finding now is that different toxins in the water can actually really impact the severity. And so what we're really hoping now is that we can use this information to, now that we know what makes their defect worse, to try and go backwards and see, can we make this defect better? And the really important thing with our work is that all the genes that we look at have been characterised in people as well. And so they've been identified in actual real patients that have mutations in these genes. Now, I've probably gone off script a little bit in answering that question, but to actually answer your question, we find that in our models, so the models we use tend to be a bit more simple than humans. And so humans, if we have a mutation in a gene, usually the defect isn't as severe as what we'll see in those simpler animal models. And I sort of liken this to having if you can imagine a football team and they do quite well and they have a star player. Depending how strong the rest of the team is, if you take that star player out, some teams will cope quite well, some teams will just fall apart completely. And so much like us, all of us, for some of us, we can remove a gene and we have a severe problem. For others, the supporting genes we have can actually compensate a lot better. So while the models we use are extremely informative, it's not exactly the same as understanding what happens in a person. But certainly what we try and do is mimic the human condition as much as possible.
Thank you, Seb. Just moving on, one of the big things that we're noting in the community is artificial intelligence. Has that had an impact on your lab, or maybe to the whole panel, whether that's had an impact in research in general?
I would say absolutely it has. And we're still very much scratching the surface of what AI is capable of. One thing that I found is it's incredibly useful for me and for my lab is sourcing information very quickly. So in the past, we'd have to do all our searches manually. So we'd have to type in, I'm looking for such and such information, because the thing is, there's hundreds of thousands of, or probably hundreds of researchers around the world working on craniofacial biology, and they publish a lot of scientific papers, they present at conferences, and often for us, we aren't across what everyone does all the time, and so trying to find out what other people have done to then try and build on their work is an essential part of our job. And AI, and even over the last sort of 6 to 12 months, the capacity to find that information even just using simple things like ChatGPT has made an incredible difference. And I really mean an incredible difference, because now I'm a lot more comfortable that I haven't missed a key piece of information or a key conference where some data was presented. And so from that point of view, it's certainly really helped the research part of my job. I mean, it's not perfect. As we all know, AI is fraught with difficulties and can make up things. But as a first pass, it's incredible. And the other thing it also does is it actually helps. And Quentin alluded to this before, for people like us, we are used to holding conversations in scientific language, so we drop scientific terminology all the time. And for us, that's our language. And so when we come and have these discussions with real people, we often find it difficult to try and say what we mean. And so what I found that things like ChatGPT can very simply translate something complicated that I'm trying to say and use terminology that is a lot more accessible. So, yes, and that's from a basic point of view for AI. And then from a more complex point of view, there's technologies that are being developed that actually help us analyse very big amounts of data. One of the things we do in my lab is we look at not just individual genes, but we look at everything that's happening in an organism. So we change one gene, and then we want to see what are all the other effects that are happening because of the one change. And so AI lets us analyse this data a lot more quickly than it ever has before. So it's an incredible technology. The biggest issue we have is knowing what technology is out there and knowing how to harness it. But it's definitely developing.
Thank you, Seb. I thought we'll just move on with Amanda, Dr. Amanda Osborne. Now, Dr. Osborne is from the University of Adelaide, and she's a clinical neuropsychologist. And previously, Amanda's team has interviewed parents of children who have craniosynostosis and other craniofacial deformities to better understand what their treatment pathway has been like. And this has been a very good focus from Professor David and Dr. McLean for many years, working out how children cope with having the diagnosis, then the surgical management, then the ongoing care of 20, 30, even 40 years. So these findings have laid the foundation for an online platform, and that's been supported by our team at Craniofacial Australia. And it provides a platform where parents and people in the craniofacial community can access and get proper research-based advice at certain times. So I'd like to invite Amanda just to explain that research and the benefits of that for us.
Absolutely. Now, is this working? Can you hear me? Okay, good. Thank you. All right. So yeah, thanks, Ben, and thanks to all of you for coming and showing your interest in Craniofacial Australia and what we do. It's appreciated and valued. So my team and I have been working with Craniofacial Australia for about 7 or 8 years now. And just to give you a bit of background, we started with looking at children with a particular type of craniosynostosis, which is where the bony plates in their skull close or fuse too early, either before birth or shortly after birth. And if the skull closes, then it makes it difficult for the brain to grow in appropriate ways. So we were interested in whether or not the functioning of these children had been affected. So we were doing some cognitive assessments and some behavioural assessments with families here in Adelaide. And the information that we found is that, in most cases, not really. Most children are doing the same as their peers at school, which is fabulous. There are some differences. There are the occasional problems with things like autism spectrum disorder. Potentially there's an increased risk there, and a little bit about the way that they process information in their brains, but generally doing about the same. So that was really interesting and informative, and it's research that we've got out to the globe in terms of this is how children are functioning. But we were doing a lot of talking with parents and families during this period because I would see them face to face and we'd have conversations about how they were going, conversations about what the journey had been like for them. And always, consistently, they were saying, we are struggling and we did struggle. And even for families that were 8, 9, 10 years down the track, they would express this feeling of, oh my goodness, where did that time go? And I was so stressed and depressed in lots of cases and worried the entire time. So we approached Craniofacial Australia and asked whether or not we could look into this a little bit more, whether we could actually learn about the experiences that they'd had and then develop some resources to help them so that the families that have children diagnosed with a craniofacial difference in the future don't go through those same stressful periods. So we were very lucky and grateful in that we were awarded that funding. And so we have spoken to parents of children with craniofacial differences over the last few years. Really in-depth online interviews because of COVID. But ones that lasted, I had one lady where we talked for an hour and 3/4 just about what she'd been through and how she'd felt and what could have helped her. So once I'd finished that initial data collection, we went through and reviewed those transcriptions from back to front multiple times, looking at what problems were consistent across these families. And there were a few things. One was a lack of information. Their parents who they're sitting in front of a GP or a paediatrician or perhaps a craniofacial surgeon, and as these guys have finished saying, lots of big words get thrown at them, words that they've never heard of, they don't understand, and they don't know where to look for information about that word. So one of the things that we've been doing is developing lots of resources that are condition specific that Craniofacial Australia will host on their website. That means that when a mum or a dad comes home from that medical appointment and they've been told that their child has Crouzon syndrome or metopic synostosis or any of those ranges of craniofacial conditions, that they can go onto their website and access some credible information. That it will be relevant, it'll be evidence-based, it will tell them a little bit about the condition itself, it will tell them what the treatment is and potential future outcomes. So along with that, there was a really big and again consistent message that the perioperative period, so just between after a child has been diagnosed, there's generally a wait, depending on what age the child is when they have surgery. But it could be months, could be a couple of weeks. And so parents are very stressed. They are uncertain about what is about to happen. And there is a lot of anxiety because after a child, they have this child, they have their baby, often it's their first baby, and they're expecting this beautiful journey and comparing, going along with friends to the park and everyone's saying, this is fantastic and amazing, and instead they're caught in this medical cycle of clinical appointments and surgical discussions, and they're on this incredible learning curve that makes it very difficult for them. And at the end of all of that, what's likely to happen is their child is going to look different. So they will take their child in for surgery on a particular day, and then they will come home and they will have a child that does not look like the child that they took in. They'll have a child whose facial structure is slightly different. They may not breastfeed in the same way. They may struggle to breastfeed. All sorts of changes. So that uncertainty fuels a lot of difficulties from a mental health and psychological perspective. Unfortunately, that then links into the fact that there are very few supports. It's not necessarily immediately offered or automatically offered by hospitals around Australia that they will be given psychological support. So many families struggle alone. Many families don't understand that other parents in the same situation are struggling in the same way. And so what we are doing is providing links, providing some fact sheets about how to talk to people about their child's condition, how to cope in those early days. The other thing is, the other theme that came out was a big discussion around peers. So incredibly important, because most families don't understand what a family who has a child with a craniofacial condition are going through. So they might come home and want to talk to their parents or their friends, and they've got no idea. And so they move online and go into online Facebook groups, for example, and get advice and support there, which is fantastic, but it has its downsides. It's quite complex in terms of the types of surgery that the children may have, the conditions they may have, the differences between the medical system in Perth versus Adelaide versus Brisbane. And so helping to link people with peers that can provide accurate information is also part of our brief. Yep.
Thank you, Amanda. You can just see the importance of supporting families at the time of diagnosis and then ongoing is essential. Just following on from that, you can see that we've made some significant contributions over the previous 5 years. If you had a magic wand or something, what would you consider to be your would be essential for the next 5 years? What aspect of mental health provision of care would be key in this area of work, not just broader mental health issues?
Yeah. So you touched on it a little bit earlier about the Craniofacial Australia Resource Hub. That's something that Craniofacial Australia's team is working on at the moment, and my team have provided a range of psychosocial, so psychological and social support, fact sheets and information that will be uploaded on that. So families anywhere around Australia can go there and get that information and assistance. Now, the thing with that though is that that's based on our interviews with parents. So we spoke with parents, we identified what they needed, we're providing what they needed. We went out to an advisory group of them and asked them, is this right? Have we got this right? And the overwhelming feedback was, yep, this is great, but add in this bit here and this bit here and this bit here. And so we modified them. And we kept modifying them based on what these parents with lived experience actually want. So they're now with Craniofacial Australia. They will be uploaded in the next few months, and that resource hub will be born, which will be very exciting. But it's based on our conversations with parents. And so the next issue is to talk to the people that actually are living with the craniofacial condition themselves. So what happened at school? What do you need there? Is it different from primary school to high school? What about when you finished and you went to university, or you started dating, or then maybe you needed to find employment, or you got married? What do you need through all of those life stages to help you get through life and deal with the issues that are thrown at you? So that's what we're doing next. Again, thanks to Craniofacial Australia, we have some funding to speak with people that have a craniofacial condition themselves. We will go through this process again, and we will update that resource hub as we go, really, in terms of this is how we can now help. So yeah, it's very exciting.
Oh, thank you, Amanda. Now, I would just open it to any questions from the floor to our researchers. Yes?
Explain the chondrocyte. I've mentioned several times.
First, I apologise for running out. I'm coming the back end of laryngitis and a bit of dry throat. So chondrocytes, you would know more as cartilage. So you know that between bone you have a soft bit, so your knees are a classic example of this. It provides a cushioning between the bones.
You said death and I thought,
Yeah, and so
death
so
and such.
so when the parts of the face form, they have a cartilage precursor, and so some bones. If you look at your leg bone during development, there'll be cartilage. So development in an embryo, this is. There'll be cartilage there, and the cartilage will be replaced by bone. In the face, what can happen, especially in the jaw, there's a cartilage element put down. So and then that gets surrounded by bone, and that's how the jaw actually forms. And so for us, if the cartilage chondrocytes don't form properly, there's not enough tissue there for the bone to grow around, hence you have structural changes. Does that make sense?
I have another question. You mentioned Professor David. For many years he was the face of Cranium. Is he still involved?
Oh, yes, definitely. He's 84 at the moment, and he's the chair of our board. And it's just interesting. All of the researchers that are looking at the micro deletions, the benchtop work, for example, and also Amanda's work are really part of what he has always spoken to us about. You know, helping families when they're in the most need. Second of all, understanding the pathology and the natural history of the disease. So many of his protocols that he's developed over the years, this is the background to it. And often research and education and training are the core. So he's heavily involved. Yeah, and he has very much supported these three researchers very positively over the years. But I used to sometimes skip through some of the articles, but he would read every word and come up with some amazing insight into how things can be practically approached. You know, what's good for the patient in the end.
Can I just add two things to that? One is that hopefully you've seen David give his talk about how the whole of the Craniofacial Centre arose and its history. If you haven't seen it, he does occasionally. Adelaide has a historical seminar series where he's given these a few times. And not to forget that in my lifetime, craniofacial surgery has been born. It's been developed from really prehistoric techniques that now are revolutionary, and so David was at the forefront of that. I mentioned in my video that we've been really successful the last few years in getting some national funding, some really a lot of funding. I met with David and Ben in the offices up on Melbourne Street probably 2 years ago. When we research, we know a lot about very, very little, right? We get so focused on a molecule. What does it do? We sometimes miss the bigger picture, right? And so that meeting I had with David that day, I walked away and I had a clear vision of what is the impact? What can we actually change with the research that we're doing? That provided me then a format to write that in a clear way so that people assessing my grants could actually understand it and understand what is the significance and the outcome of this work. So he's still providing very good insight and impact to our research, and I'm pretty sure that's true of you guys as well.
Yeah. Yes.
You talked about nutrition, diet in women. What about men prior to the pregnancy?
That's an excellent question. That's a very, very good question. And surprisingly, there's been a lot less research done on the male reproduction than the women during pregnancy. But that is certainly a very, very important factor. And there's a lot of things men can do as well pre-conception. It just hasn't been as well researched. It's a little more tricky to research that than looking at pregnancy, and sort of because there's a lot more hospital data on women during pregnancy during the entire gestation period of 9 months than there is on what the men were doing before conception. So from that point of view, it's there's just less known, but by the same token, there's probably likely to have a much greater scope for intervention during pregnancy than beforehand. And so, sorry, a lot of you probably know, we mentioned folate before as a really beneficial thing, but you'd all know about things to avoid during pregnancy: alcohol, cigarettes, soft cheeses, things like that, sushi. And that's because we know a lot of things that are harmful. So the hope is that, because we do really have 9 months to intervene pre-birth, the hope is that if we understand a lot more what can be done in the first 6 months, or the first 6 weeks rather, I should say, then I think that's really the best therapeutic window. And the thing is, in an ideal world, we'd like to tackle everything. But because, in terms of expertise and skills and just number of people working on this, we really probably have to try and focus our resources in the best, to give really the best possible outcomes, almost a low-hanging fruit where we can have an impact. But yeah, certainly we have definitely thought about, how men could, hold up their end of the bargain as well.
It's just not out there that men should be looking at themselves in this preconception stage. Yeah,
I fully agree. And I mean, you might have more.
There's also some really good studies out there, and I just want to underline this by saying I don't think any parent should feel that they've contributed to this. It's not your fault, right? If that's happened, and I know some clinicians in Sydney that often get asked that via mum. Was it my fault? Did I do something wrong? And it's not. That's not the case. And I don't want any parent to feel like that is the case. With other diseases, not necessarily craniofacial, you would have recently heard Trump now saying that autism is paracetamol's leading to this, and which probably no one believes, apart from him. And there are studies out there, and you can look at them any way you want, right? And there probably is something to do with age of father. Mother's eggs are there at birth. They don't get genetic changes. The constant, exposure to environment and over lifetimes can affect the father. But again, could that be also helped with diet? We don't know. Yeah.
Yes.
How do you change a gene? Or is it too complicated?
No, that's an excellent question. And that's actually one huge advance in technology we've had over the last 10 years. You may have heard of a technique called CRISPR, or maybe not, but CRISPR-Cas9. It's a gene modification technology that was invented, or sort of started to become popular in about 2014, and the inventors had won the Nobel Prize for developing it. And really all it is just a technique where you can very specifically make a break in the DNA, and then the cell repairs that break using its own cell machinery, but it doesn't do a good job of it, so it doesn't repair it perfectly. And so because the gene is a very, very precise sequence of DNA, if you change that sequence at all, sometimes by as much as one tiny little base pair, one tiny part of the DNA, you stop the whole gene from working. So it's a bit like if you take, now I'm no car mechanic, so I'll probably say this wrong, but if you take the alternator or the carburetor out, it's not going to work. The rest of the car's still there, but the whole thing's not going to work. Similar sort of concept with these changes. So probably in my lifetime, it's been the biggest advance in gene mutation that we can now control a lot better than we ever can. And it's become so accessible now that the students that I have in my lab, they can pick this technique up within 2-3 weeks, and then they know how to monitor the modified gene, they know how to screen for it, and then they know how to analyse the consequences. So a really massive advance. It's a great question.
Maybe we'll just have one more question.
It's not quite in relation to the studies that you were both working on, research, but CDH1 gene, can you see the connection between facial and what can be done about that? I've got 3 generations in the family that have got gene and facial.
Excellent. So CDH1 is E-cadherin, I think, if we're talking about the same gene. What I think it is.
It's definitely a cadherin.
Yeah. Yeah. Perfect. That's.
Son died of stomach cancer.
I'm very sorry to hear that. Firstly.
Both, and his sister has it and his mother had it.
So that's an interesting thing that we do find, is that there are a certain number of genes that are implicated both in craniofacial conditions and in cancer. And one of the reasons for that is that if you think about the processes of development, how Quentin mentioned this, you go from one cell to hundreds of thousands of cells to make a whole baby. In effect, cancer is a deregulation of that process. We start again with one cell and you have hundreds of thousands of cells made from that. So a lot of the genes are the same. Now with CDH1, that actually happens to be one of the targets of one of the genes that I work on. So I've had a bit of exposure to reading about it. And one of the things it does is it actually regulates the environment for the neural crest cells. So I mentioned before about you need the instructions for the neural crest cells. That's one of the genes that provides those instructions and just gives structure to the pharynx where the neural crest cells come into. So it's certainly a gene that's on my radar, and it is heavily involved with not all the processes that I work on, but certainly is quite important, particularly, and Quentin also mentioned this, the epithelium, so the lining of a lot of tissues and organs. CDH1 is important in that as well.
Can I just to finish a little bit? I mentioned my mentor Timothy Cox before, and I want you guys to leave here today knowing that these are worldwide efforts. It's not just us in Adelaide, Seb in Melbourne. I was at a conference in Italy at the start of this year where all of the neural crest geeky people were together, and we just talked shop all the time. But then Timothy Cox now runs a lab in Kansas. He was here early this year, so his fascination is on the epithelium. Have you ever seen how the embryonic structure of a face is? I don't know if you've ever looked. So essentially it's 2 halves. And they come together and fuse in the middle, right? And that happens in a lot of areas. That's why the midline defects are really common. So you have this part coming together to fuse together, and if it doesn't fuse properly, you get the cleft lip and the palate. And that is where the CDH gene is expressed in that area. So if it's not expressed properly, you get those effects. Why that then leads to a tumour, it's possibly to do with the lack of, like Seb said, the differentiation or the normal processes that would happen in maturing.
Well, we might leave it there, and let's join in thanking our researchers, and I hope you enjoyed it. Now I think we'll move on to our lab tour, and that will be led by Quentin and his team, Kay, Lee and Raf, for hosting us. Thank you very much. We would head up that way at the moment. Christine, is that right? Yeah.
What we might do is we might start here. I'll just give you a bit of an overview of what we're going to do. Is there people that don't want to come on the lab tour? Because if you don't, then I think the people that are just leaving now, did you want to leave them here? Because I'll go through introduction here as opposed to on level 10. There's more space. Yep. Is there people that don't want to go? That's a no. Excellent. So can I just bear with me for 5 minutes while I just go through a bit of an overview of where we're going? And I'll put it up on the screen. Okay, while I'm waiting for that to come up, we're going to split into two groups today. One will be taken by Kaylee, who's going to wave to everyone at the back. The other group will be with me. So I apologise now for those that come with me. That's a lie, anyway. So what I wanted to show you, Kaylee will still show the micro CT in the level 10 room. Is it that one? It's not that one. Okay, so I'm aware that probably it's the first time a lot of you have visited this building before. It's called the Bradley Building, named after one of the state's first ever Vice Chancellor women. That was Denise Bradley. It's really great that they named it after her. This is just a picture of Biomed City. I'm sure you all know where the RAH is now. Some of you might have gone to SAMRI last year. It's a beautiful building. And you would have been down the bottom of that building. The new SAMRI 2's here. We've got the Adelaide Health Medical Research at MRC, Adelaide University, and then our building just here. So this is the Bradley Building. I want to just mention quickly the Centre for Cancer Biology. Clearly we're not talking about cancer today, although we did mention it. So the Centre for Cancer Biology is where my lab is positioned, and my lab's called the Neurovascular Research Laboratory. The Centre for Cancer Biology runs from level 8 to 11 on this building. Right, you've already seen level 8's breakout spaces where we have our seminars on a almost daily basis, and then we have a lovely area and lovely views around the city. If you look out the window over here, you will see Adelaide Oval, through to the hills, through to the beach down that way. On level 12, we have dedicated animal facilities run by about 25 staff. In there, we mainly work with rodents, but we also have zebrafish. And so Seb has probably already out, but Seb mainly works with zebrafish. When he talked about the fact that within 24 hours, the head forms, that same process takes about 10 days in mice, right? So, and so we have that up on level 12. Within the building itself, every floor is very similar. If you would have seen before, out to this side are the lab spaces. Here we have open office areas, and then we usually have more dedicated offices down the other side. We'll see that when we go up to level 10. Today I'm going to be taking you through level 10 mainly, that's where my laboratory is, and then also into level 9 as well. I'm aware that there'll be some, if you can't take steps, we'll always take elevators, that's fine. And myself leading one group and Kaylee the other. So neurovascular research is what we do in my lab. This is the lab. We've got about 12 people at the moment. Yep, that's 12. All right. And there's others that come in and out with that area. I just wanted to also say that the vast majority of people in my lab work on that stem cell we talked about before, was the neural crest cells. They are the foundations for the bone, the cartilage of the face. They form all of the peripheral nervous system. Any pigment that you have in your body comes from those same cells. They just give rise to such an enormous amount of mature cell types that are necessary for us to exist. So what do we do? We're really interested in understanding is how do childhood defects arise, right? I'm sure you all recognise the picture on the right, Treacher Collins Syndrome, from a very famous movie called Wonder. And this is a speciality of the clinical team here in Adelaide, as is craniosynostosis, which you would have heard about before. On the left-hand side, there's two different types of cancers. One is neuroblastoma and another one is a brain cancer. So these are the types of disorders we work on, and we really want to understand how they arise so we can try and stop them. How we do that is to try and understand how embryonic development occurs. On the left-hand side, these are all human embryo pictures. Left-hand side is a single cell. The oocyte becomes fertilised. It goes through a rapid series of transitions to give rise to something that's relatively normal within 23 weeks of gestation, right? After that period, we just have really growth and some maturation of cells. So how do we study that? It is hard, right? A lot of the stuff we use are cells, and Seb mentioned before some really current technologies, which was CRISPR, but another Nobel Prize came about recently for induced pluripotent stem cells, and this was Yamanaka who won that. Yamanaka found that you could take skin cells, we can take it from any one of you, we can take a cell from you, we can add some growth factors to it and make it now a stem cell which could form any cell type. So with a range of factors, we can take these stem cells and form any of the organs. I mentioned before I was at a conference in Italy just recently, and they're now taking these stem cells and can start to make jaws. They can start to make facial features. So we're at the cutting edge of what we're about to do, and if we can do that in a dish, that'll be heaps easier for us, right? And we can answer more questions, ask different questions that we can do with whole animals. So one of our toolkits is stem cells, but also cells in general. Another toolkit we have is mouse embryos, right? I said a mouse house upstairs. Mice are very good because if you look at the pictures from human on top to mouse on the bottom, they look very similar. The mouse has all of the same things we have. They're 91% similar at the genetic level, right? They form the same structures at the same times. So if we understand in mice how it happens, we understand how it happens in humans. Even the heart in mice is the same four-chambered heart. It still has all the same things. Whereas in other organisms, that's not the same. But we also use fish. And so this is a picture of 12 hours of fish development. Now, what's the benefit of fish is they lay eggs. You can watch all of this outside of the mum and in real time. And this is just a very quick picture of what we can do if it's going to play. It's not going to play. I'm going to show you that picture later when we're up in level 10. It's open on level 10. And so what you'll see in this period, there's 2 cells just here. Within 12 hours, that's giving rise to a beating heart and blood flowing around, and the craniofacial elements are starting to form, right? And you'll see some videos as we walk around about that. So what are we going to see today? As we go through, we want to show you some of the cells, some of the chondrocytes that we mentioned before that form the cartilage. We're going to look at stem cells. We're going to look at neural crest cells. These are elements, precursors for all the cells we talked about. We're going to look at differentiated stem cells. And some of the technologies we're going to use there range from like quite gross microscopes where you can just look at it with yourself. We'll go to 10 times the size of what it actually is, through to other microscopes where we look at 100 times magnification. So when you look under microscopes, and I'm sure you've all seen a microscope where you look down the little lens pieces, they can range from, like very small amounts of magnification through to really high levels of magnification. So what might you look at there? So under low magnification, you can look at a whole organism. I can see a limb, I can see a jaw, I can see teeth. When you get high magnification, we're looking at a single cell. We're looking at molecules, separate proteins within the cell. We're looking inside the nucleus, right? You'd have to guess that there's an element of cost that comes with changing there, right? The low-end microscopes are $10,000. The high-end microscope, which you'll see today, it's an individual room, it's $1 million. I'm not saying for a second that we buy that ourselves. It's a joint piece of equipment that 500 people use, right? So we can reduce costs by many people and multi-purpose. We're going to show you some whole organisms. In this case, Seb mentioned something called micrognathia, and that just means a small jaw. We're going to see that under